Welcome to the International e-workshop on wastewater-based COVID-19 surveillance. Despite the different time zones and your busy schedule, especially those who are heavily involved in the surveillance task, I thank you for making time to join us in this workshop. Of course, we know some cities are still in the midst of lockdown, while some have seen improvements in the pandemic situation. However, we still should not let our guard down. Surveillance monitoring must still be in place to ensure continued security against the spread of COVID-19. As an environmental scientist and engineer, it is heartening that we can contribute to this pandemic through wastewater surveillance. The objective of this workshop is to allow us to come together to share and learn from each other the experiences, knowledge learned from the conduct of wastewater surveillance, and hopefully we can enhance the wastewater surveillance approach. I would like to thank the supporting staff from NERI for their hard work over the past 1 month in swiftly putting together this 2 days program. Thank you to our speakers for accepting our invitation and all of you for participating in this e-workshop. Joining us today are almost 500 participants from 42 countries from various professions. Before we proceed further, let me give you a very brief overview of this 2 days e-workshop program. Today, we shall start off with a keynote delivered by Professor Joan, a world-renowned expert in water microbiology, water quality, and public health safety. In 2020, Professor Rose led the International Water Association Task Force on COVID-19. I will share further details of the vast credentials later in the program. After Prof. Rose's keynote, we will have 8 speakers from India, Singapore, South Africa, the Netherlands, and USA to share their experience through the implementation of wastewater-based COVID-19 surveillance in their respective countries. For tomorrow, we have dialogue sessions covering planning and sampling, handling and analysis, and data analysis and reporting. These dialogue sessions allow us to dive deep to discuss the various aspects of implementation, execution, to decision-making, and communication in the surveillance program. I hope we have an enriching and fruitful session in these coming 2 days. Thank you. Okay, next, I'd like to invite distinguished Professor Tsuhan Chen, NUS Deputy President of Research and Technology, to give us an opening remarks. Professor Chen, please.
Thank you, Prof. Ng. So, like how Yong said, good evening, good morning, and good afternoon to all of you. Thank you very, very much, yes, for joining us at this 2-day international workshop on wastewater-based COVID-19 surveillance, organized by NUS Environmental Research Institute, NERI. Warmest welcome to all of you, the participants, whether you're in Singapore or overseas, for joining us online. Say the least, COVID-19 has certainly affected everyone since early 2020. It's changed our lifestyles and many other things. The number of COVID-19 cases has since then reached more than 140 million and caused over 2.5 million deaths to date. After more than 1 full year already, we're still fighting and defending our life against this pandemic. Wastewater-based surveillance, as many of you are the domain experts in, is a technique that's used in assessing public health and infectious disease. It has been shown to provide early detection of COVID-19 infection within the community. This method, many of you know already, is done by screening wastewater samples of SARS-CoV-2, the virus that causes COVID-19. SARS-CoV-2 can be detected in feces of symptomatic or asymptomatic individuals. Hence, wastewater COVID-19 surveillance could capture both types of infections to complement clinical testing in mitigating the spread of this disease within the community. I'm very, very glad in going through the program to see that my colleagues in the NERI will be able to share with you how they have conducted surveillance here on our US campus and how this tool is important to community living, such as our own school hostels, but also at workers' dormitories. I'm very excited to see the program and to even more so look forward to learning from all of you, the domain experts from all over the world, to share with each other your expertise and experiences. So finally, on behalf of the National University of Singapore, I'm deeply honored to be able to bring together all of you, the experts, the practitioners from different parts of the world, to this work. Sincerely, that through this workshop, you could share the knowledge and bring new discoveries to wastewater-based.
Is there any, Prof. Chen? I think we couldn't hear you. I think we are losing you, Prof. Chen.
Can you hear me at all?
Now okay, now okay. Yeah.
Okay, so how much did you get? No worries.
I think the last part. Yeah, now we can hear you clearly. Please continue. Yeah.
Oh, I just finished.
All right, Prof. Chen, thank you very much. Thanks. All right. Okay, now we shall start the technical session. I'll be your chair moderator for today's sessions. Okay, before we begin, there are some simple instructions to all the participants. Angel, can you share the screen? Okay. So first of all, Angel, can you off the sound? Yeah, okay. So this webinar is being recorded and the recordings will be available on NERI website after this workshop. Now, during the speaker presentation or during the Q&A sessions, if you have any question, post your question in the Q&A icon located at the bottom of your Zoom panel. Okay. Now, if your microphone has been muted, that's during the Q&A, and the chat function has been disabled. If you wish to speak out loud, you may click on the raise hand icon located at the bottom of the Zoom panel. If time permits, we will unmute your microphone for you to speak during the Q&A sessions. Now, if you see any question posted by other participants, if you have similar or same question, you can upvote that question by clicking the thumbs up icon, okay? And we will address the most upvoted question first. All right, we may not have enough time to answer all questions. So what we are going to do is that we're going to compile this question and bring over to tomorrow's dialogue sessions to continue answering your questions. All right, so that will be the arrangement. All right. Okay, the first talk we have is a keynote presentation by Professor John Rose. The title of the talk is The Evolution of Environmental Virology. Let me introduce Professor John Rose. She holds the Homer Nowling Chair in Water Research at Michigan State University. She's a member of the National Academy of Engineering and the winner of the 2016 Stockholm Water Prize. In 2015, she was awarded honorary citizenship of Singapore for distinguished contributions to the country's development of sustainable safe water. She's a member of the Royal Institution of Singapore. She is an international expert in water microbiology, water quality, and public health safety. She has published more than 300 manuscripts. She led the International Water Association Task Force on COVID-19 in 2020. She is also in charge of the Global Water Pathogens Project. She's currently a member of the IWA Board of Directors. She's currently collaborating with the state of Michigan on the statewide program on wastewater surveillance for COVID-19. All right, without further ado, may I invite Professor John Rose to share your screen and present your keynote speech.
Thank you very much. All right. Okay, I might have to go to the other share screen. Sorry about that. There we go. Now I should be able to move it. Yeah, there we go. Okay, so I wish I was there in person. Singapore's my second home, and I haven't visited for quite a while, as we're all just staying home, of course. So I thought I'd give a brief overview of the evolution of environmental virology from where we were and where we are now with SARS-CoV-2. I think all of us here are dealing with viruses, and I want to say hello to the global community that is joining, this seminar, and thank NERI and NUS for putting it together. This really is a phenomenon. I mean, we used to have to kind of introduce viruses, but now people know what a virus is and they see it on the news. And we do know that viruses infect all living things, that they're quite small, they're nano bioparticles. In environmental health, we're used to dealing with a list of about 100 different kinds of viruses, but mostly enterics. Those were viruses that infect the gastrointestinal tract and then infect other parts of the body, but are excreted in the feces. So and they're spread through the fecal oral route. So those were the viruses we were used to evaluating in environmental health area. But through history, it really has been this sort of link between health, technology, and science that has influenced where we are today. So what happens in medicine, what happens in our technology, and what happens in science. Some of the first descriptions of viruses were poliovirus in the 1700s, the clinical description of the disease. Yellow fever came later and it was later identified as a virus and was a key environmental virus spread through mosquitoes. By 1892, we knew there was infectious, filterable agents, there's contagion. The early 1900s, the vaccinia was cultured so that they were looking at how they could develop a vaccine for smallpox. Bacteriophage were discovered in 1915. This led to a lot of understanding of host virus interactions and also, of course, the understanding of DNA. By 1930, yellow fever and smallpox were actually cultured. So cell culture came along at that time, but it wasn't really perfected for some of the viruses of concern in wastewater or the fecal oral viruses such as polio till about the 1950s. In the late 1950s, we started using the electron microscope. And by 1985, 35 years later, we had the ability to actually look at the DNA using PCR, polymerase chain reaction. I want to introduce in terms of the history two viruses, hepatitis A and polio. And if we think about hepatitis A, we know from historical writings that what they called epidemic jaundice and liver disease were around as long as humans were around and they could document these things. In particular, what they called campaign jaundice was recognized within military troops. And of course, in the US, we saw this in the Civil War, it was recorded, and in the First World War. In the 1940s, the disease was named infectious hepatitis, and they could distinguish it from, the blood-borne hepatitis. And it was actually detected under the microscope in the early 70s. And there's a photo there of some of this scientists looking at this, using the electron microscope to look at viruses. And it took almost 5 decades though. So 50 years later, even though they had studies on immunology to develop a successful and efficient vaccine for hepatitis A. And we still have a fairly broad global burden of disease of hepatitis A despite having a vaccine. And so we see some parallels here, maybe not the speed in which we can gain knowledge, but this issue of understanding the disease, its transmission, and the development of vaccines. And yet we still have this issue, even in the United States, where we've seen cases in all age groups come down due to sanitation and due to the vaccination. We've had a major outbreak that in 2017 that actually started in California, swept over the United States, hit Michigan particularly hard, and hit the homeless. where we have vulnerable, sensitive people that were dying of hepatitis A. I bring to your attention Irene Zaragarki's work published about that time as well. And she was looking at the surveillance of hepatitis A virus using wastewater and using these new techniques, as well as other enteric viruses. Now, poliovirus is also an interesting one. If we look at it, again, it was the disease was depicted early in paintings, the paralytic impacts. It was fully described by Sir Walter Scott, an author. And there was a very broad clinical description, and it was called infantile paralytic disease because it did seem to affect children more than anyone else. But epidemics began to appear really in the 1900s, and it was identified that it was contagious. As I've mentioned, it was cultured in primates first in the 30s, and then in cell culture in the 40s and 50s. It wasn't actually until 1941 that they identified that this virus grew in the intestinal tract and that it was actually a fecal oral pathogen. And you may know some of the history of the vaccine programs, which began with very broad campaign. There was a live vaccine, the Cutter incident, where individuals had a reaction to the vaccine. They stopped the vaccination program and had to regroup. We now have a global polio laboratory network that is run by the WHO. This consists of 146 accredited laboratories in 92 countries and these regions that are evaluating polio in wastewater. And in fact, they're able to look at wild type and the vaccine-derived poliovirus cases, which started emerging. And so we have this look of where we're monitoring in other places where we've eradicated the disease. And again, while poliovirus has been eradicated in these continents, except for Asia, Afghanistan, and Pakistan in particular, sewage surveillance played a very vital role in looking at both wild type vaccines, if the live vaccine was being used, and these variants. And we've heard about variants showing up before. And so again, it was sanitation, this whole idea of public health measures, as well as vaccination programs and surveillance that has led us to this point where a very serious illness is now, we're at the brink of completely eradicating it. Now, we've got hundreds of pathogens, of course, found in excreta and sewage. And the early studies on sanitation and disease, where wastewater and surveillance and epidemiology was kind of brought together, was in Feacham's book. That was one of the first, where they talked about bringing these three disciplines together. And this book has been redone, Sanitation and Disease in the 21st Century, and is available on the Global Water Pathogens Project website, waterpathogens.org. Now, the GWPP started in 2014, and it's built an online platform. Right now, it's got this book, which has 115 chapters, a lot of resources and tables and data sets. The network has grown to about 250 people from about 52 countries. The section on viruses has 9 chapters right now, and everything adeno, hepatitis A, hepatitis E, the norovirus, papillomas, of course, poliovirus, the polyomaviruses, rotavirus, astroviruses, and sort of a summary of emerging issues. The chapters are set up to look at disease burden, transmission, a brief section on detection methods. It's not a methodology book, but it's really focused on what do we know about quantitative information about viruses in wastewater, their persistence and their removal particularly by wastewater treatment processes. And I give you some of the levels, the gene copies. Of course, as this book got redone, the scientists that were redoing these chapters really focused in on the use of a polymerase chain reaction, PCR, for quantifying the virus in wastewater. And so you can see that we found levels up to as high as 10 to the 7th gene copies per 100 mils in raw sewage. Now, we have a snapshot of viruses around the world, like rotavirus, which is an important virus, and there are campaigns to try to implement and invest in the rotavirus vaccine, particularly for young children. But it's a snapshot in time and space. So you just get a look here and there about the percent positive samples at different wastewater plants and the concentrations. Same thing with some of the other viruses. And in some cases, we don't really even have a very large global database on these viruses. And here comes Coronaviridae. So this family of viruses has been known for some time. It's a positive sense, single-stranded RNA virus, very much like poliovirus in that sense. But of course, it's different in that it contains an envelope with these glycoproteins. And we've all seen the virus now on television. It's found in humans and animals, the range of viruses in this family, 200 different hosts. And in fact, the first coronavirus was identified in 1932. It includes the common colds. This family includes common cold, SARS, MERS, and I think 2012, the outbreak in MERS lasted about 4 months. In the SARS outbreak lasted, of course, there's still cases, lasted about 6 months, but these outbreak situations. SARS hit the scene, of course, very, very rapidly. And it was very early on that we understood that this virus moved to many organs in the body, away from the nasopharynx, the throat, the stool, the sputum, and that it was able to replicate in cells in the intestinal tract. It was detected in feces, and it wasn't long after that that the techniques that have been used for studying viruses in wastewater began to be explored for this particular virus, both the concentration and detection methods, the primer sets that were being used in the clinical world, the gene sequencing, particularly for gene targets like N1 and N2 genes, were developed. And early on in the pandemic, it was recognized that this virus could be found in sewage. We've got these terms now that we're using. Sewage epidemiology was an old term that was used, and now we're using this term wastewater-based epidemiology. But it's quite interesting because if you Google that term, wastewater-based epidemiology, FEACHM, as I mentioned, was one of the first references that shows up, kind of merging these terminologies. But actually, sewage epidemiology was a term that was used between 2005 and 2011 to describe the study of drugs and pharmaceuticals in sewage, and to look at the community use of these drugs and community health. This term morphed into wastewater-based epidemiology and was being used by 2015 for this type of surveillance. There were special issues. The American Chemical Society had special sessions on using this analysis for studying mostly some biomarkers, but mostly drugs in the community, drug use in the community. And we now see this use of this term merged with the environmental virology history and the COVID-19 wastewater-based epidemiology around the world. I just want to tell you real briefly about what we've been doing in Michigan. We have a large network of about 20 laboratories, and doing some 200 locations using the same methodology to explore SARS-CoV in wastewater. This happened very quickly. In Michigan, we started looking at the methodologies around April 2020. And by May, there were several labs using some of the methods, exploring the methods. And basically, after this, in this midsummer and late summer, the funding went out to support this laboratory network. And this network was already using quantitative PCR, qPCR platforms for microbial source tracking markers and E. coli on beaches. And in this case, they decided to move from the quantitative platform to digital. So all the labs were able to buy an upgrade to a droplet digital PCR platform. And if you know anything about the digital platforms, this uses instead of a standard curve to quantify your target, it uses a most probable number by distributing the sample out into multiple droplets and then quantifying presence absence in each of these droplets, and then providing a quantitative estimate based on a most probable number approach. The digital worked out very well. There was very rapid learning and standardization, good quality control using the digital platform. There seemed to be less interferences with untreated wastewater as your matrix in using the digital platform. And so that's what MSU is using. And this is how the drops, you can see the droplets right now with the positive and the negative. And then there's always this kind of uncertain range in terms of what's positive and what's negative. This area in here, this middle range, is sort of important because we're starting to see these variants, PCR platforms for variants. And they tend to be showing up in this high range, but at very low droplet numbers. So Michigan is like some part of the rest of the United States. We had this peak in 2020 in March and April. You can see the daily cases, and of course our deaths were quite a concern during this time, the April to May timeframe. It came down during the summer. There was social distancing. The state of Michigan shut down, which was quite controversial. And things seemed to be going a little bit better, but it started seeing a rise in October. And of course, this second wave is quite high. The wastewater signal, as we started with the various communities, and this is April and May, was also high in most of the communities. In some of the communities, you can see that it came down in the summer, down to 0 non-detect, some of our smaller communities. With some of our larger communities, we still saw some of these little peaks. It was still seemed to be positive here and there. We saw some outbreaks associated with bar openings in the summer and things like that. Now, what's been happening since is that, of course, we've seen it rise again from our from the summer and into this October timeframe that I just mentioned. But we are starting to see in communities this January and February, it start to come down in some communities to non-detects. We've got other communities where we're still seeing some cases and some large peaks of the virus. In Michigan, we were doing both N1, we were doing 3 genes at one time, N1, N2, and E gene, but the decision was made for the network to stop doing the E gene. It did correlate, but it was less efficient, and the N2 and N1 are quite highly correlated and give us confidence of the occurrence. We're also doing surveillance on the campus at dorms. That was quite challenging to set up. And of course, as you can imagine, we have several teams that go out during the winter, brushing off the snow and lifting up the manhole, getting the samples. And we have moved from the fall semester, which was a logistics, a development of our logistics, our methodology to try to decrease the timing from the sample collection to the results, using automatic samplers now. This is sort of what we're seeing in this spring semester, I guess you would say. There was one dorm that was a little bit high when they moved in, which was not good. But we saw over time that all the dorms started to show. So this is time on the x-axis and space on the y-axis here. And we saw some very high peaks by the end of January. These are starting to come down, and we're starting to see some negatives, but there's some residual virus levels. One of the things we've done in this case is we have both the wastewater results, which are this top row. The black are the positive, the orange are the negative. And then we have the saliva results in the same places. And we have the surveillance results, which are when they test somebody that's sick or test somebody that has been in contact with someone. So the blue is what they called someone that had come in contact, so they're quarantined, and the red is actually someone that's isolated because they tested positive. We're now analyzing this data now, but you can see that what the wastewater signal was high before we started seeing some of these peaks. And it matches pretty well with the saliva work, which was measuring both asymptomatic and symptomatic, whereas our bottom row was measuring symptomatic individuals as well as those that had come in contact or could have possibly turned positive. So this data is being analyzed now to see if we can get a better handle on calibration of the wastewater signal to the cases, and also the duration of excretion, and our hypothesis that early in the infection, you get high rates of excretion. So we're evaluating that now. So you all are here and know why we're monitoring wastewater. And we want to use this information as a leading indicator in some places, early warning. We want to be able to demonstrate the relationship between existing diagnostic testing and wastewater surveillance so that it's useful to guiding our resources from the public health as we move out into the community. We think that spatial risk mapping might be very useful, information to help support rapid testing and screening tactics. Of course, the vulnerable populations are important, and nursing homes and correctional facilities and other places where we've got areas where they could, where the risk could be high. And of course, the sequencing of and tracking of new variants in the community. We are, the waterpathogens.org is now taking on another project. We do have the book. We also have tools there for mapping and for enteric virus mapping and for looking at virus pathogen removal through wastewater treatment. We're now developing a site on COVID-19 called Wastewater Sphere, the SARS Public Health Environmental Response Program. Dr. Medema, who's one of your speakers here, has been key to helping develop this, this database. We're working with Dr. Colleen Naughton from the University of California, Merced. You've probably seen her website where she's been inventorying the monitoring of SARS-CoV-2 in wastewater around the world. We now know that, and this was a couple of days ago when I grabbed this screenshot, there were 56 dashboards, 233 universities involved around the world in monitoring either their own campus or helping monitoring the community. It's 50 countries and 1,000, almost 1,400 sites around the world. So this is quite a unique phenomenon compared to previous surveillance in that it's spatially very broad. There's some consistency in methodologies, and we've got this temporal agreement in how we're monitoring around the world. The WSphere mission is really to advance environmental surveillance of sewage, and we want to inform public health measures. We hope to integrate data across various field deployments and dashboards The dashboards are mostly in high-income countries. We're starting to see a few emerge in other places. And of course, we want to be able to visualize the data sets and also provide access to the scientific community to the global data sets. More importantly, we're interested in use case studies, which shows how wastewater surveillance is used in public health response. This is a collaboration between PATH, a nonprofit organization out of Seattle that works in developing regions of the world on public health, Michigan State University, KWR, University of California, Merced, and Venthic Technologies. So we are starting to look at global maps. We've got a platform that's a demo platform. It's not gone live yet. We're starting to download data sets from various places in the world. This is Scotland's. So you can get a global view of where there's actual quantitative data, how much there is. You can then focus in on the country or focus in on the actual locations and the data and be able to actually visualize the data in terms of trends, up or down, and access the actual quantitative information. We're developing online submission forms, and there will be curating of the information. So we hope to have something rolling, that's visible to the public in the next 2 months. I want to end just by saying that we've got these new variants. I think wastewater surveillance is going to be very important. It's a different gene. Of course, it's the spike protein. And there's a lot of new variants, but of course, they're concerned with those in which there's been a change in infectivity, which appears to be, from what I can tell from the literature, high excretion rates. It may be excreting a little bit higher in some cases. There's been some evidence of that, but it's not, that information is not widespread. We don't know that all the different variants are excreted in the feces at the same level. But also there's evidence that in epidemiological studies, it's transmitted faster, that are not. And in cell culture, it has greater affinity for some of the receptors. There have been some PCR assays developed by a variety of companies now. One in particular that's being used in the US gets at the UK variant, but also a gene sequence that kind of represents the genes captured in the UK, South African, and Brazilian mutations. And just recently in Texas, they found about 33 out of 35 wastewater facilities around Houston had the new variant. They didn't talk about quantitation, like how widespread is it, but it's there, at least in the wastewater. So I want to thank you for inviting me here. I'm looking forward to the session. It's very early in the morning, so I can get my coffee now and sit back and listen to the rest of the experts. I also want to acknowledge all the individuals that I work with in the state, Dr. Shannon Briggs, who runs the state agency, and of course, our Rose group and the Michigan State group that is looking at doing all this work. So I'm going to stop sharing here.
All right.
And I think that I did okay on time.
Yes, you are way ahead of the time. All right, thank you, Prof. Rose. In fact, we have two questions for you. First is, what action did you take if a dormitory is in the red area?
Yeah, so the wastewater surveillance was not leading the actions on campus. It was more confirmatory. So as especially in the fall semester, it led to more of the saliva testing because that was not mandatory at that time. So when the spring semester, when the students moved in the spring semester, they said now it's mandatory for the saliva testing. And we're going to have some wastewater evaluation. We're going to let you know if there's positives. And so the students, of course, don't want to be isolated. They don't want to be quarantined, of course. But if they're sick, the University Health Sciences Center goes in to help get them tested with the nasal pharyngeal test, the PCR test, and then they support them through isolation. But at MSU, we're not moving the students. So we're not moving them, we're supporting them in place. Whereas other universities, they're moving them. We also then, we're able to then broadly apply the saliva test. And the hope is by the fall semester is that we only use wastewater and then move into the other more extensive testing as the wastewater signal shows up. And we're hoping that our wastewater signal can be 0. But interestingly enough, you know, there's a lot of students that live off campus, and we monitor one site of the sewer in that campus site, and they've had quite high levels bouncing around. So the off-campus students who are going to be coming to campus are also going to be, we hope to bring them into the communication strategy and also set up areas for testing. Of course, ultimately, we want to vaccinate. And faculty and staff, especially older faculty and staff, those that have pre-existing conditions, and then provide opportunities for our students ultimately to get vaccinated.
Great. Yeah. Okay, the next question we have is, there has been discussion about sampling wastewater from locations where there are small number of people contributing in terms of ethical issues without consent. What are your thoughts?
Yeah, so it's kind of interesting when you go into that wastewater-based epidemiology and sewage epidemiology literature on monitoring wastewater from a community or from a certain area in a city. There were quite a few articles on the ethical issues associated with taking samples and looking at community health through this window. And what does that mean in terms of this consideration? And especially as you go closer and closer to a location, which we're doing now, like a nursing home, a specific nursing home. So I think the ethical issues are in regard to communication. And at the wastewater plant, communicating broadly with what's going on. We've had some new variants show up at our wastewater plants in Michigan as well. They're very, they're there, but they're there in very small concentrations. Only a few droplets are positive. And they've been communicating that in the newspaper to the broader community. Now, at the nursing home or correctional facility where they're doing monitoring there, that is being kept confidential. That is not being put up on a dashboard or any of that. That is confidential. It's being communicated back to the facility so that the facility can take action, and the communication can go to the residents so that they know what's going on. And so, yes, I think there's some ethical issues as we get closer and closer to where we're monitoring. And there's going to be some data that needs to be kept confidential.
Great, thank you. More questions for Prof. Rose. Next one, which groups? Where can we find more information regarding the variant PCR assays?
Well, there's two companies that we're working with now, and I haven't explored all the different kits. We do have a team, part of the network in Michigan is looking at these different kits. So GT Molecular has a kit. I believe that it uses, and that's the one we're using, the one I just showed you, gets the UK variant and then the other 3. And that's a PCR primer-based, and it works with both qPCR and digital. BioRad also has some primer sets that works with their digital droplet machine, which we have. We haven't tested their kit yet. We're going to be looking at that. And our guess is that there's going to be other companies that are going to come forward with primer sets now, and maybe kits that allow us to look at these variants using our PCR methodologies. I think some of these kits may be very platform specific, but we're hoping that most of them will be generalized so that we could use them across various platforms. So GT Molecular is the one that we've been using thus far for evaluation.
Okay. Joan, there's a follow-up question. How are you discerning the variant droplets for the non-variant droplets when you analyze your PCR data or sequence?
Yeah, so the digital platform that we use is, can do, and we've been using it for source tracking and other things. So you have two types of fluorescence, and so the droplets show up in different fluorescent markers. So, and I didn't produce a slide, but then you get a quadrant of how many droplets are positive for one fluorescence marker, how many droplets are positive for the other fluorescent marker, how many are positive for both, and then how many are negative. And so you get a view using digital droplet. And so it works quite well where you run the assay together using the digital system. The other digital platforms that are out there that are not droplet based also have this capability of looking at more than one marker with more than one fluorescent channel, so to speak.
Okay, thank you. Maybe a last final question. I think this question should be pretty straightforward. Are there any successful examples worldwide for implementing sewage for identifying hidden patients to help control the COVID-19 spread?
So I think there's quite a few examples now, around the world we're seeing. Australia at the country level, Australia is using presence absence. I know Dr. Gershon Medema has been looking at this, and we may have some participants from Australia on this webinar, and so that's been used quite successfully. At the University of Arizona, of course, they've gotten quite a lot of attention because they used it as early warning, and they moved students out of the dorms to an isolation dorm, so a dorm that was designed just for students that had turned positive. And they were able to show that they could detect early on that there was infection, go in and do testing, move and isolate, and stop the spread. And that was the key thing. Now, I think it's going to be interesting to see what is happening with the vaccination program, because we still have some cities we're monitoring that were quite high. And the larger cities, the signal is still there, even with closing down the city and the public health measures. Now they're starting to implement the vaccination program. And what happens then to the signal as we get higher and higher percent vaccination in the large city, the small city? And so I think these use case studies are going to be quite interesting and maybe guide us as we say, are we reaching everybody we need to reach, especially the underserved in the vaccination program? Because we know in the United States, we're not doing a good job of that. The vulnerable are falling behind in terms of getting vaccinated. And just like we saw with hepatitis A, that's where we're going to still have problems.
Great, Professor Rose, thank you very much. Thanks.
Thank you
for an interesting talk and the reply to all the interesting questions. We look forward to more new findings from you, especially with the vaccination, what happened to the signature in the sewer. Thanks. All right.
Thank you.
Now we move on to the next speaker. The next speaker is Professor David Larsen from the United States. His talk is Leveraging Wastewater Surveillance to Improve Health Security. Dr. David Larsen is an epidemiologist at Syracuse University with expertise in global health. Most of his career has been focused on the surveillance, control, and elimination of malaria, while also adapting and applying those principles to other infectious disease systems. Dr. Larsen is now an associate professor in the Department of Public Health at Syracuse University. Throughout the pandemic, Dr. Larsen has advised on coronavirus surveillance and control at Syracuse University and led initiatives to establish a wastewater surveillance platform for the state of New York. All right, let's welcome Dr. David Larsen.
Thank you. Thank you so much for having me today. And thank you, Professor Rose, for your talk to introduce the topic. It's very much a pleasure to be here and present some of the work that we've been doing I'm one person of a very large team, and so I will come through and present this. So I'm an epidemiologist by training, and really focus on elimination of malaria is where I did a lot of work, and knowing that to eliminate a pathogen or know that it's gone from an area requires a great deal of data and a great deal of infectious disease surveillance. And so, you know, in the initial stage of the pandemic, I watched with interest what was happening in Wuhan, and then Italy. And by the time before it hit New York, I realized that the same problems that I had been dealing with in malaria were problems in coronavirus pandemic, and particularly in the United States. And so I transitioned some of my work over to that, and identifying, I think Dr. Medema showed the proof of concept that the wastewater could be used, you could isolate virus from wastewater. And so we began initiatives in April to try to scale the statewide platform in New York. And I'll go through that today. And so I want to put the wastewater surveillance in the context of what was happening early in the pandemic in the state of New York in March and in April of 2020. This figure here shows the number of diagnostic tests conducted each day in the state of New York. We suspect that coronavirus arrived on American shores. We know that it arrived by at least January 2020, and there's some hints that it may have arrived earlier. And it spread relatively quickly. In New York, we had a statewide school closure by executive order of the governor on March 18th. And we were doing fewer than 10,000 tests, diagnostic tests per day. You know, that stands in contrast to today where, you know, earlier in just a month ago, we hit 250,000 diagnostic tests per day. One of the reasons we were not doing these tests is that there were strict guidelines on who could be tested. So in the county that I lived in, a person could not even get a diagnostic test unless they had contact with someone who had traveled from China. And we now know that, you know, China was not the primary source of coronavirus in New York State. The New York State was seeded most likely from Italy. And so we have this big disruptive public health intervention that was enacted in the absence of complete absence of understanding of where coronavirus was and where it was not. If we look at the state, you know, New York City is down here in this top left graph in this little cluster of cases. This is New York City, and there's a very large state behind it, right? Very rural state of New York. And in March, you know, you had clustered transmission in New York City. In April, it kind of spread to Long Island, Westchester counties, a little bit further out. By May, it hit the primary urban centers of New York City. Each of these orange counties are urban centers. On the far left, the west part of the state, you have Buffalo, and then you have Rochester. This orange one in the middle right here is Onondaga County, where I live, and then Albany County is right here. And this is the major industrial cities where you see more transmission, and then other areas, these rural areas, very little transmission, and that's May of last year. And then by June, we had a little outbreak at a strawberry farm in one county, and it was starting to calm down. But the fundamental principle here is that low transmission, infectious disease transmission is very spatially clustered. And as transmission increases, it spreads out geographically. What that means for public health is at low transmission, you can be very precise with your interventions. You can precisely target areas, geographic areas where transmission exists. And then as transmission increases, as the pandemic spreads, you lose that ability for precision in responding to something like the coronavirus. And so I guess this is kind of the thesis of the talk, or the theme of the talk is wastewater surveillance could have been instrumental. You know, we were doing maybe 10,000 tests a day in April. The genetic sequence was released in 2019, December 31st of 2019. The proof of concept from Medema was in March. Had we scaled up wastewater surveillance at that point across these about 614, I think, treatment plants in the state of New York, we could have had much better definition of where transmission was and where it was not. Oops. Let's see. And so our team uses an UltraSucrose method, is kind of what we termed it. We tried a few different methods that weren't quite working at the low transmission we were observing. And this is outlined in Dr. Hyatt Green's preprint here. It's under peer review right now. But the idea is we take a 24-hour composite, we load it into an ultracentrifuge, and importantly, they add this sucrose cushion that acts as a filter. We had tried using some commercial filters, but we couldn't get them in stock because of supply chain disruption. And really, the sucrose was supply chain resistant. And so in a pandemic, I think we've all faced supply chain issues. So the sucrose syrup method worked really well. It was done at Upstate Medical University. The Syracuse University wouldn't let us bring any wastewater on campus, nor would SUNY ESF, which was quite interesting. There was a lot of fear about transmission of virus, which we don't expect to be relevant. But working in Dr. Frank Middleton's lab in the first parts, and then doing quantitative PCR in the second part. And I'm going to focus on the math and the surveillance around the data outputs. I keep pushing over, sorry. All right. And so we worked in about 14 counties, this data over the course of last year, 14 counties, 24 different treatment plants, and about 76 different sewer sheds. It doesn't include New York City. New York City has a wastewater surveillance program. The state of New York is kind of interesting, where they have a Department of Health for the state, and then the city runs its own Department of Health as well, and they're kind of separated that way. And so the first question, as we got going, is I'm most interested in the negative results. From a public health perspective, the absence of an infectious disease is our deliverable. It's what we want to do, is to make sure that the disease is not there. And so can we first leverage these negative results to ascertain freedom from SARS-CoV-2 transmission? And so we take the geospatially matched confirmed COVID-19 cases to sewer shed, and then we quantify our wastewater results into 3 different levels, right? So the level that they're not detected, there's no indication from the analysis that SARS-2 RNA was found. And that's a function of how much wastewater you analyze, the sensitivity of the method as well, how much interference from the matrix. And then so there's not detected. Then we have this category where it's detected, but it's at such low levels that it's below the level of quantification. And my microbiology colleagues, they cringe a little bit at this, and maybe Professor Rose and some others are cringing here. But I'll show you what this means from a freedom from disease standpoint. And then we have the quantifiable results, where it is above that threshold. And then we use these freedom from disease principles to estimate absence, control of transmission, absence or control. And these are derived from agriculture, from veterinary science. And when bringing animals to market, the need to ensure that that you're not bringing infectious disease with that herd into a market setting. And then we could apply these, and these have been used for malaria and some other pathogens as well in elimination settings. So when we get our results, I'll focus on the top 2. Is we have these 3 different categories: quantifiable, detected, and not detected. And the top left here is the COVID-19 incidence in the sewer shed. And we start to see very visible clustering of when it's not detected, clustering around 0 cases per 10,000 population. When it is detected, a little bit higher, and then the quantifiable, of course, it can extend beyond with limitless bound there, or bound at the number of population, right? And so we start to see this differentiation in 3 different categories, and the same thing for test positivity. When it's not detected, 1 day following, test positivity is quite low. When it's detected but not quantifiable, a little bit higher, and then when it's quantifiable, a little bit higher. Now, of interesting note, we're using two messy data streams. We're here, so it's a bit limited. All these quantifiable results where there's no detection of cases, I consider that just the limitations of using health system data, where we know that not everybody has symptoms from coronavirus, not everybody seeks treatment, and then not everybody can get a test. And so there's that. And so these are less concerning. More concerning from a freedom from disease standpoint is this group here of where we did see cases that came from a sewer shed where it was not quantifiable the preceding day. But then again, these cases, we don't know precisely where the transmission event occurred, or we don't know necessarily the physical location of the toilets they're using. We did do our best with the data. And so we're pretty comfortable in saying that we think it works. There's clear differentiation. When we look at the proportion of results and how many cases the day following, this purplish bluish shaded area is the negative, the non-detection. And we see a little bit of overlap here, but quite a bit of, this area here where it's fewer than one case per 10,000 population here, and then it works. It looks pretty good, in my opinion. And when we look at some scenarios, then we say that if we detect a non-detection, we have about 30% of the time we had 0 cases over the next week. And then 70% of the time we had fewer than 2 cases per 10,000 population. And so that gives us a measure of sensitivity of the wastewater surveillance to input into our freedom from transmission principles. And we can actually start modeling out the probability that a sewer shed or a community has no coronavirus transmission. And I say that no coronavirus transmission in quotations because we know from a public health perspective, we're only concerned about coronavirus transmission if it reaches that outbreak stage, right? If we have an imported case that doesn't go anywhere, then it's totally fine. Or if we have a little chain of transmission that's cleaned up, then it's okay from a public health standpoint. We don't have these aggressive measures. But when it reaches that outbreak stage, that's where it's problematic. And so this here on the left is 2 different counties where we start modeling the probability that the transmission is under control. And with each negative result, this probability increases, right? And these correspond here to the same number of cases across the entire county, right? And so here we're having 1 case every other day, a couple 2 cases, and then notice, I'll show you what happens in not Cayuga County, I'll show you what happens in Cortland County later. But once you do detect the virus in the wastewater, then you see that then your confidence that there's no transmission in that community declines. And so these two would be used in conjunction with each other, the case data as well as the wastewater data. In Cortland County, we see the same thing. We see this increase in likelihood of no transmission, and then it drops out because we see a detection that corresponds with a little cluster of cases right here, and then that cluster is cleaned up, and we start to see again an increase in the probability that transmission is under control. And then it's fine until it drops out again, and that corresponds to this cluster of cases over here. So the second question, beyond giving that confidence that transmission is in control or under control or absent, then can we ask, well, does it provide an early warning system? And so within our 14 counties, we had 3 outbreaks that made the newspaper. And they were in 3 communities that had state university campuses. And so we matched the timing of the first detection of the wastewater to the detection or the quantification of we matched timing of the detection of coronavirus in wastewater to the beginning of the community outbreak. And each of these counties had minimal detection before. And so we started a little bit late with these counties. They started in August as opposed to some of our other counties started in June. And so we saw low detection here in Cortland County. I guess Cortland County actually started in June, so maybe I should extend this graph. But Oswego and Oneonta started later in Otsego County. And then we saw this early warning that something's been detected, and then it jumps to quantifiable. And that corresponds to a cluster outbreak of cases at the college, right? So courses begin on September 1st, or just before September 1st. We start detecting. All the students came back to SUNY Cortland. And then we see that it jumps up to quantifiable in the community. And these are actually community cases, not college campus cases here as a cluster. And then this outbreak actually was quite extended throughout the fall of 2020. In Oswego County, at the west side, the treatment plant west side of the river, where the campus is located, we saw a little bit of fluctuation in detection, non-detection, before it jumped to quantifiable levels. And you see here the cases in the county is a little bit of fluctuation, and then it jumps again up here with quantification. In Otsego County at SUNY Oneonta, they began and they had a little bit of non-detection and detection. They actually responded with public health measures, as they had planned to test their entire student body like 1 month into the semester, and they actually moved that up to do it as quickly as possible based on this wastewater result. Came back down to non-detected and then jumped up again to quantifiable, coinciding with a really large outbreak that shut down the university. It was something like 200 cases over 1 weekend in a single university. And so there is some indication that the wastewater surveillance can provide an early warning of an outbreak in these communities. And it's about 1 week in terms of the date of the wastewater sample and the date that the clinical test was conducted. And then the early warning is actually greater. I apologize, I keep doing that. I push the over arrow. Is greater if there's delays in the testing. Then the next question we can ask is, does wastewater surveillance correlate with transmission? And again, we use these geospatially matched confirmed COVID-19 cases at sewer shed. And we utilize a measure of intensity for our SARS-2 RNA, where we divide our number of copies identified in the wastewater by the number of copies of crassphage DNA. And we've played with crassphage DNA and the crassphage RNA. Crassphage is a ubiquitous bacteriophage that exists in human guts, and it's been pretty stable over time in our wastewater surveillance. For the non-detections, we quantify that as 1 copy, and for the detections, we quantify it as 3.5 copies. Our limit of detection is 5 copies per milliliter of wastewater. And then we use 1 copy for the non-detection because we log transform this, so that turns it into 0. And then we just estimate the correlation. And again, we're using a messy data stream with the health facility cases. And so we use moving averages with a 1-day window, a 3-day window, and a 5-day window. And we look at incidence and test positivity. And across these 76 different sewer sheds, we see correlations are pretty good, about 0.55. They're not nearly as high as been observed at some of the major metropolitan areas where they have daily samples from a single treatment plant serving the entire metropolitan area. But the highest correlation we observe here is with this 5-day moving average, appears to be the best. And here at 1 day of lead time between wastewater and clinical cases. But that correlation stays moderate up to 9 days out. And then beyond that, we don't expect biological or public health. It just doesn't make sense to be forecasting beyond 9 days. Right, and so the best correlation observed with only 1 day lead time, and that's between the wastewater sampling date and the date of the clinical test. Now, clinical tests can take time, right? It can take a few days to come back, and so as can wastewater. And so if you can engineer the wastewater testing to be quicker than the clinical testing, which I think is possible, then you can have even more lead time, and then you do correlate pretty well going forward. And we're still working on modeling, moving this one forward. Then the next question, well, what is the cost of a statewide wastewater surveillance program? And so I just input some estimates that we had for our proposals, compared to those to New York State budget and tax revenue figures, and then estimates from population from the U.S. Census Bureau. All right, and what we use is about a $350 per sample for a centralized program. That includes program administration as well as data modeling and the wastewater testing. In the United States, I've seen a range of prices. At the low end, I've seen as low as $150 for a wastewater test, and at the high end, $1,200 for a wastewater test. And we use this 300 measure, try to return results within 36 hours. And I don't think anybody needs to be paying more than $500 per sample, which has been previously marketed. And so for an entire year of wastewater surveillance across the state of New York, the estimate would be about $23 million for twice weekly at 632 municipal plants. And to put that into context, we had the CARES Act in the United States. New York received something like $6.5 billion in CARES Act, the state of New York, for the Department of Health. And for this information, we're talking less than 1% of the CARES Act to scale up a wastewater surveillance platform across the state of New York for an entire year. There's also been estimates of how much state sales tax has been lost. And so we have here, it's about, the cost of this program is about 3% of the lost sales tax revenue during the pandemic. And in terms of affordability, I think it's pretty clear how affordable something like this is. And Professor Rose touched on the variance. Mathematically, which is true. We can detect variance in wastewater, and I personally see wastewater surveillance working in partnership with whole genome sequencing of clinical samples, where the whole genome sequencing of clinical samples identifies mutations of concern, such as the 6970S deletion. And then wastewater surveillance is the most cost-effective way to map out and monitor the geographic extent of a variant or a mutation of concern. And so in this top figure here is the incidence from January 17th to 24th of this year throughout New York State. You can see it's much more widespread than it was early in the pandemic. And I focus on non-detection of the pathogen. And so if we do not detect a variant of concern or a mutation of concern, what does that actually mean? And so we can take the inverse of the transmission intensity in a county or in a sewer shed and give us a threshold for variant detection, right? If we're confident that we can detect about 1 case per 10,000 population, then using that inverse, we are confident that we can have about 10 to 35% as a threshold that we know that a mutation of concern is below that if it's not detected. And we increase that confidence with repeated non-detections. Now, if we do detect it and quantify it, then we can just take the ratio of the variant of concern to the standard test. And so, in conclusion, wastewater surveillance can rapidly and cost-effectively measure community-level transmission at wide geographic scales. It's a type of environmental surveillance, where from an infectious disease surveillance standpoint, if we can get information on where a pathogen is without touching humans, it makes it so much more attractive. Because with humans, you have access, you have more ethics, you have increased costs as it's one-to-one. And so we try to pull these measures of the environment, such as wastewater. And the two big things, in my opinion, are the first, the confirming that it's absent or control, or the transmission is control. All right, this allows for much more precise public health interventions. That school shut down on March 18th. To me, it was the right choice given the information we had at the time in March of 2020. But had we had wastewater surveillance at that time, we could have had a much more precise intervention. We could have shut down specific school districts. We could have curtailed movement in specific locations as opposed to a broad statewide measure. And we need to move our health system to do that. And I think it's a no-brainer in terms of cost relative to budgets, at least in the state of New York. And I view it as a critical element of health security. That our health security was breached with the pandemic in New York. Our way of life was greatly disrupted, and so we need to establish systems and institutions that preserve this security, so that we can live a more free life and enjoy freedom. And a big thank you. We had funding from a couple university seed grants, New York State, and the municipal wastewater treatment plants, and again a big team here. And then if anybody's in the New York State area or the Northeast area, we do offer this services. And our industry partners, Quadrant Biosciences, that commercialize the UltraSucrose method, and then Arcadis that we worked with on the New York State pilot. Thanks.
All right, thank you, Professor Larsen. Due to time constraint, maybe just one question, and we'll leave We have a total of 5 questions from the participants. We shall answer one question and we'll keep it the rest for tomorrow. Okay. So the first question is, how were limits of detection and quantification defined? All right. Just now you show a curve, right? How do you set the limits of detection and quantification?
That's a good question. So I'm an epidemiologist, and I don't define limits of detection in terms of copies per milliliter. My microbiologist colleagues do, and the limit of quantification for our method was 5 copies per milliliter of wastewater, and but I think in terms of limits of detection as how many cases per population can we find. And that's the human relevance, and are we missing cases? And so our limits of detection, when I look at it, it's about 1 case per 10,000 population is what we were observing. And to put that in context, there's a great write-up it was actually NPR. I don't know if it's been peer reviewed quite yet, but to maintain coronavirus under control, you have to be able to maintain contact tracing, right? So if you don't want the pathogen to spread, you don't want a broad community shutdown, then you have to isolate chains of transmission through contact tracing and interrupt that way. And as you reach 1 per 10,000, that's probably manageable with the health department, with the local health department, and able to do that. As you exceed that, you get 2, 3, 4, even 5 cases per 10,000, and then you're just going to be overwhelmed. That contact tracing, and either the virus is just going to spread, or you have to have more population-wide interventions.
All right. Thank you, Professor Larsen.
Thanks so much.
Keep the question for tomorrow. All right, let's move on to the next speakers. The next speaker is Professor Anna Maria de Roda Husman. She's the head of the environmental department at the RIVM National Institute for Public Health and the Environment Center for Infectious Disease Control. She holds a chair in Global Changes and Environmentally Transmitted Infectious Diseases at the Institute for Risk Assessment Science at Utrecht University. And the title of her talk is Setting Up a National Sewage Surveillance for COVID-19 and Beyond. All right, so may I invite Professor Husman?
Thank you very much, Professor Ng. Can you hear me well?
Yes, we hear you very clearly.
Thank you very much for inviting me.
Yeah,
yes, and I have to agree with Professor Rose that it's such a pity not to be able to be in Singapore at this moment, because I also love to come in and visit your university.
When the travels is allowed, we'll certainly get together, hopefully by next year.
Yes. Yes, hopefully. And hopefully sewage surveillance can contribute to that. So I hope you can now see my screen.
Yes.
Yes, and my presentation.
Yep.
That's great. Okay. So setting up a national sewage surveillance in the Netherlands has been quite interesting and also quite a challenge. And what I would like to discuss with you today is actually how we did it, how we were challenged, and how we managed to be able to contribute something to COVID-19 preparedness and response. So our location is actually in the center of the Netherlands, and what we have been doing at our location is collecting sewage samples from a large area of the Netherlands. And what I would like to do is to go through milestones, challenges, and to see what's beyond the sewage surveillance for COVID-19. So the core tasks of our institute, which is the National Institute for Public Health and Environment, is actually to advise the national government and other authorities, professionals, and citizens. We implement prevention programs. We monitor the health, diseases, and the quality of the living environment. We contribute to prevention, control, and management of incidents and crises. And we conduct scientific research and collect and interpret and share this available scientific knowledge. So what we have been doing from the very start of the introduction of SARS coronavirus 2 in the world is seeing whether we can use the sewage water research as a tool to identify the virus early and to see whether this is a good addition to national COVID-19 disease surveillance. We receive and analyze samples twice a week at this time from over 300 sewage treatment plants in the Netherlands, which I'm going to show you shortly. And that means that's approximately sewage derived from 17 million people. The calculations that we do obtain a number of virus particles in the sewage water per 100,000 inhabitants, which allows for comparisons between municipalities and regions, but also between this indicator, these sewage figures, and other indicators such as positive tests or hospitalizations. This national sewage surveillance, of course, is not something that we can do by ourselves. So we set up a tripartite, which we put down into an agreement for the coming years. And this is between the Dutch water authorities and their union, the National Institute for Public Health and the Environment, where I am, and the Dutch Ministry of Health. But besides that, we also have a lot of help from water laboratories, engineering agencies. We work together with this Foundation for Applied Water Research. We work together with the Dutch Ministry for Infrastructure and Health, so another ministry besides the Ministry of Health. Of course, we work with the municipal health services, which are very important to us, and I will show you that in a minute. We work with the scientific community as yourselves, but also with the Central Bureau of Statistics in the Netherlands and international agencies such as WHO and EC. What we do with our numbers is actually put them on this Dutch Ministry of Health coronavirus dashboard. And I put up the way how to reach that. And this data is then used to inform people about how to act, which measures are in place. And fortunately, our sewage data have been there for quite some time now, I think half a year. It's been a challenge because people keep asking us, what does it mean? Can you explain peaks? Can you explain trends? And it's all still in development, but we also, as a public health institute, think that it's worthwhile and it's important to put our data as open data. So at our website, you can also find all the sewage data that we gather and the numbers of virus particles in the sewage as open data. So I think you lost me at the local decision support, which is very important actually. So can you hear me and can you see my screen?
Yes, we hear you and
thank you. Thank you. So I'll continue. So what's very important is how to use these signals in the sewage to be able to promote a local follow-up of municipal health services together with the municipality and the water board. We actually produced a fact sheet on the sewage figures that you can see on our governmental Corona dashboard to help people understand these figures. And at this moment, we're developing the decision tree in which we develop signal values that are then decided upon by policymakers, which should be the signal values to support local action by infection prevention professionals in municipal health services. So this is coming up, and we'll of course translate it into English as well. So what are the milestones in developing our national program? First of all, scaling up the sewage surveillance from 1 to all of those 315 municipal sewage treatment plants in 6 months has been quite a milestone for us. We are very fortunate because most people in the Netherlands are connected to the sewer lines. So that means that we can collect the sewage from almost all of those 17 million Dutch inhabitants. What's also been a milestone is setting up quantification of SARS coronavirus 2 RNA in sewage. It's one of the few it's one of the few exact numbers that are indicators for COVID-19. Furthermore, we supply open data on our website, and we deliver the data actually in a very short turnaround time to this Corona dashboard. What has also been a challenge is the geographical translation of sewage treatment plants. We actually managed last week on the 4th of March. So on the Corona dashboard at this time, what that means is that the safety regions and municipalities are exactly coded to the sewage treatment plants, which are, of course, different areas. We have been communicating within and outside the Netherlands on setting up this program and also building capacity internationally. We have established a method to identify variants of concern. And also we found wide support for the national program, including the broadening of the scope, especially with the water boards. So I'll show you a little bit more about that in a minute. On the right hand, you can see how the sewage treatment plants are very clearly differing in size. So we have the larger one in the west of the Netherlands, where more people are living close together. Whereas in the north and in the east, there's clearly smaller sewage treatment plants. So challenges which we encountered in setting up this program were to establish a signal value in relation to other indicators. This has not been easy, especially with those differences between the first wave, the second wave, and the third wave, which currently seems to be moving on. Also in quantifying current and emergent variants of concern, I saw there's more people thinking about this. It's much more easy in human samples to quantify the level of the variants of concern, such as the UK variant in relation to other variants in the sewage sample, because of course variants are originating from many different people in the sewage. Governance has been challenging. That's because of the large diversity in stakeholders. So you see people from the engineering community, from government and government agencies, but also infectious disease experts, and you have to explain what sewage actually is and that it contains feces. Also, scaling up and intensifying the program during a pandemic has been quite challenging because, of course, everything is at a distance. So when you're hiring new people, you will hardly meet them because everybody is working online except the people in the laboratory, of course. And then you have this challenge of the modelers who are working at home and the laboratory people who, of course, mostly work from the laboratory. So it's quite a challenge. Furthermore, setting up a tripartite and a big data flow has been quite a challenge. So combining laboratory and metadata that you need for your calculations of the sewage figures. And then there's the ethical considerations. And I would like to take a very short while to get into that. So we were triggered or inspired by our Canadian colleagues who came up with a very good document, Steve Rudy and colleagues, on ethics and communication guidance for wastewater surveillance. And what we've done in the Netherlands is for our sewage surveillance program is put out a request for ethical approval. And we got word back, official wording that we do not require approval under the Dutch law for medical scientific research on people, which has been a very well-received message. And also, we think, as was mentioned before, that it's quite a challenge when you talk about smaller locations. So we now also requested ethical approval for locations such as schools, nursing homes, hospitals, where we also perform research in the sewage. Future challenges, I think mainly the disentanglement of contrasting trends. So what you see is more and more people are becoming vaccinated, but variants of concern are coming up, and so they are more prominent. They may give us higher virus loads. I didn't see anything yet on them being more prominent in the feces, but I'm sure research will pop up. Measures such as a lockdown also influence the sewage load, and then there's also behavior. So whether people have themselves tested, whether they adhere to interventions that are taken under lockdown. So understanding the virus load in sewage lately has become more challenging. And then, of course, keep the momentum. So in a post-COVID time, let's see how we can keep the attention to the sewage surveillance. At least we have the wide support of the Ministry of Health, the water boards, their union, and the municipal health services. They support us widely. And what we have been coming up is thinking about sewage surveillance beyond COVID-19. So, of course, this is not new. There's a lot you can research in the sewage. You can look at hormones, you can look at drugs, you can look at other viruses or other pathogens. We are exploring at the moment whether we can broaden our program to other parameters. And we started off with this very nice Dutch chart, which you probably do not understand. I'm sorry for that, but it also has some nice pictures. So you can see how the human biomonitoring from sewage coming from urine or feces can help us understand the local or regional health of the population. So with respect to capacity building, we've been receiving many bilateral requests, invitations to webinars, protocols, consumables, helping out with guidance documents with the WHO. So has been a lot going on, and we've been trying our best to help and answer any questions to help other people also setting up their national program. And last but not least, of course, there would be no sewage surveillance program if we wouldn't have our water boards that are constantly supporting us. And the Union of Water Boards, specifically Mark van der Werf, Peter Verlaan, Meindert Spallebroek, have been supporting us in quite a tremendous way. The Ministry of Health, the Ministry of Infrastructure and Water, Municipal Health Services, water laboratories, and other institutions, and of course our own team at RIVM, which actually consists of many people, but especially Willemijn Lodder and others who've been helping out, and Jerome Locke with the international collaboration. So from my side, this is it, and I'm ready to answer any questions.
Thank you, Professor Husman. All right, we have two questions here. First is acknowledging RIVM was a leader for polio environmental surveillance. How has the experience for SARS-CoV-2 wastewater surveillance compared?
Well, actually, in the Netherlands, we are monitoring for polio in sewage pits. So it's quite different from the sewage treatment plants that collect a lot of sewage water from inhabitants. And this is done mostly at schools and, for instance, migrant locations. So that's a bit different. But on the other hand, we have been learning a lot, of course, about how they take the water samples, how they concentrate their sample, and how they detect the virus in the sewage. Yes.
Great. Next question, how do you manage to quantify variants of concern? Do you identify new ones?
Well, new ones, not yet. We're just following, I guess, like everyone else, following the emergence of new variants closely. At the moment, we have identified UK variants, but also South African, Brazilian variants of concern, like everybody else. And we find this quantification quite challenging. I saw from colleagues that they've already managed, but of course the viruses come from many individuals that add their feces to the sewage. So actually we think it's quite optimistic. There's some form of quantification that we can do, but let's discuss that also with our colleague who just presented his results. Yeah.
Okay. So Singyot, do you have Singyot, you have a question, is it? You raise your hand. Singyot, are you going to ask a question? or you press accidentally? Okay. All right. Okay. So once again, thank you, Professor Husman. Let's move on to the next speaker. All right. The next speaker.
Thank you very much.